Okay, today we are going over the cell respiration worksheet as a review for your test. So let's get everything set up here. Here we go, guys. If I go too fast, just ask me to slow down. Let's begin. Write the equation for cell respiration. 6 moles of CO2 react with 6 moles of, I am doing photosynthesis. My bad. 1 mole of glucose plus 6 moles of oxygen yield 6 moles of CO2, 6 moles of H2O, and ATP. You don't necessarily have to say 38 ATP. ATP will be fine. Yes. You turned it online. Well, how do you want to? Huh? Yeah. Now you're on my YouTube video. That was Denislav, everybody. Interrupting the video. You're watching YouTube. That was Denislav. Denislav Yordanov. Is that how I pronounce it? Okay. He's the interrupter if you wanted to know. You good? All right. X is a reducing agent. It becomes oxidized. It lost its electron. Y is the oxidizing agent. It becomes reduced. It gains the electron. In cellular respiration, the conversion of glucose and oxygen to carbon dioxide and water, which molecule is reduced? What is the final electron acceptor? Oxygen. What molecule is oxidized? Glucose. C6H12O6. What happens to the energy that is released in this redox reaction? It is stored in ATP bonds. If you just put ATP, that's fine, but if you want to get really specific, it's in the bonds. When you store energy within bonds, that's going to be like potential energy. Then when you break the bonds, energy is released. Yeah, Drew.
Does it kind of just like sit lower and be stored? Is that what happens?
Say what?
Does it like just the ATP? It's like stored in ATP.
That's fine. It's stored in ATP. Yeah. NAD plus is called an electron carrier. Its reduced form is NADH. Fill in the tally of maximum ATP yield from this oxidation of 1 molecule of glucose to 6 molecules of carbon dioxide. Initial phosphorylation of glucose, -2. Remember, initial glucose is being phosphorylated. It's gaining phosphate. ATP is losing them. Substrate level phosphorylation and glycolysis, that's +4. But I want to make sure you all understand that's a +2 net. When you lose 2 and you gain 4, that's +2 overall. Oh, instead of saying overall, we say net. In the Krebs cycle, it's +2. In oxidative phosphorylation, it's +34. So the maximum total is +38. How much ATP can be generated by how much more can be generated by respiration than by fermentation? Respiration produces 36 more ATP. Think about it. Glycolysis leads to fermentation. Can anybody remember how much ATP is made net in glycolysis? 2. So that's just 2 without oxygen. You can make 38 with it. 38 minus 2 is 36. You make 36 more. Here's why. O2 is needed for the ETC. The ETC is aerobic. It requires oxygen. It produces the bulk of ATP. Out of the 38 total ATP that are made in cell respiration, 34 of them are made by the electron transport chain. And you need oxygen to run that chain. All right, now we're going to get really specific here. Complete the table below. Main function, inputs and outputs. The main function, break glucose. Inputs. All right, here we go. Glucose, 2 NAD plus, 2 ATP, 4 ADP. Outputs, 2 pyruvate, 2 NADH, 2 ADP, 4 ATP. And if you want to add those 2 waters, fine. No harm, no foul. 2 waters are made, but it's not a big deal. You good? Okay, let's go back up to the top of the other column. All right, that is the prep reaction. The function is to metabolize pyruvate to acetyl coenzyme A. The inputs is acetyl, actually 2 of them, 2 coenzyme A's, 2 NAD pluses. The outputs are going to be, 2 acetyl coenzyme A's, 2 NADHs, and 2 CO2s. Yeah. Oh, you're right. I don't know why. Again, a little groggy here. You're right. Instead of 2 acetyls, it should be 2 pyruvates. Thank you. Yeah, help me out, guys. It's been a long journey yesterday. Citric acid cycle. Let's see. The purpose is to metabolize carbon molecules to make a lot of electron carriers. So the inputs to acetyl, 10 NAD pluses, 2 FADs, and 2 ADPs. The outputs, 4 CO2, 10 NADH, 2 FADH2, and 2 ATP. Oxidative phosphorylation. This is the electron transport chain. The majority of the energy in all of cell respiration is from here. 34 out of 38 ATP are made here. So we have 10 NADH, 2 FADH2. Oxygen, if you want to put 34 ADP, nothing wrong with that. Okay, and then fermentation. Let's see. Metabolize sugar in the absence of oxygen. Remember, glycolysis will come first, and then you go to fermentation. So, guys, over here in this final box, it depends on what type of fermentation you're doing, whether lactic acid or alcohol fermentation. So I'm going to separate them too, but the inputs are the same. 2 pyruvate, 2 NADH. Now, if you're doing lactic acid fermentation, which is what I will put on top, your 2 products will be 2 lactate, which is the same thing as lactic acid, and 2 NAD plus. if you are going to perform alcohol fermentation, which things like yeast will do, you are going to produce 2 ethanol, which is the type of alcohol, 2 CO2s, and 2 NAD pluses. Any questions with the table? Okay. All right. Number 8 through 20, multiple choice. One thing I'm going to tell you as we get closer and closer to the AP exam is when you take a multiple choice test, if you're not sure what the answer is, cross out the ones you know it isn't. It improves your odds, X percentage more. A substrate that is phosphorylated has lost the phosphate group. Nope. Has increased reactivity. Nope. Has been oxidized. Nope. That's with electrons. We'll pass this electrons to the electron. Nope. That's electrons. It's B. It's formed by the reaction ADP plus P yields ATP. It's a phosphate. If you don't know what the answer is, get rid of what you know it isn't. Number 9. Which of the following is not true of oxidative phosphorylation? It produces approximately 3 ATP. Okay, that is true. So I'm going to put check. That's true. Actually, hold on. Yeah, it involves the redox reactions of the electron transport chain. That is true. It involves an ATP synthase locate. That is true. It uses oxygen as the initial. Is that true? No. It is an example of chemiosmosis. That is true. So D is the correct answer because it says which one of these is not true. There are questions like that in the AP exam. They even capitalize not. You got to watch out for stuff like that. My last period, someone go, oh, I thought it said true. You got to watch out for that. Number 10, substrate level phosphorylation involves a shifting of a phosphate group from ATP to a substrate. No, it's the other way around. That's not it. Can use NADH or FADH2? Nope, doesn't need it. Takes place in the cytosol. Well, actually, maybe. That might be it. Accounts for 10% of the ATP formed by fermentation. Nope. Is the energy source for facultative anaerobes? Oh, yeah. That's the one. Now, I want you guys to key in on this. Anaerobes. An anaerobe is an organism, mostly bacteria. You tell me, do they or do they not need oxygen? They do not. Under anaerobic conditions. A facultative anaerobe can make its own ATP without oxygen. And that process is substrate level phosphorylation. Okay, let's go down to 11 through 20. All right, glycolysis spelled wrong, no big deal. The major reason that glycolysis is not as energy productive as respiration is that, let's see, I'm reading them right now. Pyruvate is more reduced. Nope. It does not take place in a specialized membrane-bound organelle. That's not why. C is a fact, but that's not the reason. It is a pathway common to fermentation and respiration. That's not, I mean, that's a fact, but it's not the reason. NAD plus is regenerated by alcohol or lactic without the high energy electrons. It's E. Substrate level phosphorylation is not as energy efficient as oxidative. And you don't make all that. You only make 4 ATP from substrate level. You make 34 from oxidative. Number 12, you guys should be able to get this. Which one is it? C would be correct. Here's why. You don't make CO2 in glycolysis, so A is wrong. Acetate, nope. You don't make that much ATP in glycolysis, and you don't have FADH2 in glycolysis. C is the correct answer. Process of elimination. Number 13, when pyruvate is converted to acetyl coenzyme A, CO2 and pyruvate are released. That is a fact. Let's see what the others look like. A multi-enzyme complex removes a carboxyl group, transfers electrons to NAD+, and attaches a coenzyme. That actually happens too. Oh, wait, whoa. A is wrong. Sorry. That's wrong because it says pyruvate are released. That's not true. So A is definitely wrong. Wrong. B looks good. One turn off of the citric. Nope. NAD plus is regenerated. Nope. No. The answer for 13 is B. Number 14. Which of the following produces the most ATP per gram? Do you remember? Fat. Definitely fat. Fat is a lot of energy in fat. That's why when you fast, the first thing your body's going to target is your fat. And that's going to go through all that because that's the best source. I've seen on the news that there's Yellowstone or Yosemite or some national park had this fat bear competition. And Otis the bear won the contest for the fattest bear. The bears right now are getting really fat because we're getting into winter in a couple months and they're going to be hibernating because there'll be no food. So they're not going to go out in the elements. Fats and proteins can be used as fuel in the cell because they can be converted to intermediates of glycolysis in the citric acid cycle. 15 is B. 16. Which of the following is not true? The enzyme phosphofructokinase. You guys did not have to know this. The answer is D. You probably Googled it, but you did not have to know that one. So if that totally startled you, don't worry about it. Cyanide is a poison that blocks the passage of electrons along the electron transport chain. Which of the following is a metabolic effect of this poison? So back during the Nazi days, whenever they had Nazi spies, if a spy was captured, they all had like a drilled hole in their tooth and they would hide a cyanide pill in there. And if they ever got captured, rather than be tortured and forced to give secrets of the Nazi regime, they would loosen the pill at their tongue, stomp on it, and die. Like in Wonder Woman or like Captain America, they showed in those movies. The pH of the intermediate space is much lower than the normal. No. Alcohol build up, and it is also E should be the correct answer. All right, number 18. Why is glycolysis considered one of the first metabolic pathways to have evolved? Guys, what happened on Earth first? Life or the presence of oxygen in the atmosphere? Who says life? Who says oxygen? It's life. There was life on Earth before there was actual oxygen in the air. So life had to figure out how to survive. I shouldn't say figure out. It had to evolve to survive without O2. The answer is E. Sure. Yeah. Number 19. The metabolic function of fermentation is to... it's definitely not E. E is just silly. There is no electron transport chain, so B is gone. Oxidize pyruvate in order to release more energy. Oxidize, so they're going to call us A. A is the correct answer. And number 20. What is the role of oxygen in cell respiration? I've seen this question on every test I've taken and every test I've given. What is it? D. It is the final electron acceptor. All right, so there is your cell respiration paper. You guys may hold on to that until Thursday and you can turn them in. You can use this as a study guide. Let's switch over to the photosynthesis one now, if you can pull that out. Here it is. We have about 17 minutes, so let's get to it. What occurs in the process of photosynthesis? Give me one moment to get my pen ready. All right, here we go. Light energy is converted into chemical energy. So energy from the sun is going to convert that into things like sugar. Write the overall equation for photosynthesis using words. Carbon dioxide and water react with sunlight to produce glucose and oxygen. If I go too fast, just say something. Write the overall equation for photosynthesis using chemical formulas. 6 moles of CO2 react with 6 moles of water in sunlight to yield 1 mole of glucose and 6 moles of oxygen. What does photosynthesis require in addition to water and carbon dioxide? Sunlight. What is the principal pigment of plants? Chlorophyll. And when I was up in Wisconsin, I could see some trees were starting to change color. They don't change color as much as like Virginia and the northeastern states, like the New England states. But I thought of you guys on a Saturday standing around Wisconsin. I took a photo of the sky for you. Here it is. Why are the plants shedding their leaves? Do you see sunlight? I don't. It can be cloudy like this for months. No sunlight. You get into the winter. Is there any liquid water? No. So here's the cause and effect. This time of year, the xylem cuts off. Result, no more water flow. Result of that, no more chlorophyll. So what color will the plants not be anymore? They won't be green. Do you remember what that pigment is that they use as a backup to chlorophyll? Carotenoids. What colors can they reflect? Orange, red, and yellow. Eventually that runs out. The leaf dies. Is the tree dead? No. In fact, I tried to get a photo of a tree. Yeah, here we go. Is that tree dead? That tree is not dead. It will regrow its leaves in the spring once the clouds dissipate and the snow melts. All right, circle the letter of the region of the visible spectrum in which chlorophyll absorbs light very well. Which colors are absorbed very well? Blue and red. Yellow, a little bit of yellow, definitely not green. Number 8, chloroplasts contain sac-like photosynthetic pigments called thylakoids. What is a granum? Stack of thylakoids. The region outside the thylakoid membranes in the chloroplast is called the stroma. Careful not to get that confused with stoma. What are the 2 stages of photosynthesis called? Light reactions and Calvin cycle. Complete the illustration of the overview of photosynthesis by writing the products and the reactants in the process, as well as the energy source that excites the electrons. So what gas is going, or not necessarily gas, what 2 reactants are entering the light reactions? Water and sunlight. Is CO2 used in the light reactions? Definitely not. What comes out? Oxygen. What goes from the light reactions to the Calvin cycle? ATP, NADPH. This is the Calvin cycle. What goes from the Calvin cycle back to the light reactions? ADP, NADP plus. Someone in here is going to make the mistake by saying photosynthesis has NADH and cell respiration is NADPH. Pretend the P means photosynthesis. Someone's going to make the mistake. I'm going to say, I warned you, I told you, and you're going to say, yeah, you did. So careful. CO2 enters, and you guys could have put a few things here, but actually these 2 boxes should have been the same. G3P is the product, and that will make glucose. Somebody sent me a screenshot, and they said, I selected glucose for this question, what is the product of photosynthesis? And I put glucose, and it was wrong. And so, well, it's actually G3P. That's what comes out of it. How many G3P does it take to make 1 glucose? 2. And you could put a few things in these boxes. The one that's on the diagram above, it says sucrose, which is a disaccharide. It's called table sugar. And then you have starch, which is also amylose, which is a big polysaccharide stored for energy for later use. All right, circle the letter of the carrier molecule involved in photosynthesis. B, NADP plus. How does NADP plus become NADPH? It is reduced by 2 electrons. Those electrons came from water. Those electrons will go into photosystem 1, and then it'll make NADPH. All right, Calvin cycle. Actually, no, not yet. Not yet. Circle the letter of each sentence that is true about the light-dependent reactions. Guys, the word light-dependent reactions is another name for light reactions. There's really no difference. It's just another word. Correct the false statements. They convert ADP into ATP. True or false? True. They produce oxygen gas. True. They convert oxygen into CO2. It converts H2O into what? Oxygen. The oxygen that we inhale came from water thanks to autotrophs. They convert NADP plus into NADPH. Correct. Where do light-dependent reactions take place? Thylakoid membrane. What happens to water in the light-dependent reactions? It is broken into oxygen, hydrogen ions, and electrons. Circle the letter of each sentence that is true about the light-dependent reactions. Correct the false statements. High-energy electrons move through the electron transport chain from photosystem 2 to photosystem 1. Is that correct? Photosynthesis begins when pigments in photosystem 1 absorb light. What is it? Photosystem 2. The difference in charges across the thylakoid membrane provides the energy to bank ATP. Yes. What are the products of the light-dependent reactions? Here we go. ATP, NADPH, and O2. The light energy from the sun is converted to what type of energy is stored where? Chemical energy, which is the ATP. It's stored in the bonds. Whenever you break a bond, it releases energy. How does ATP synthase produce ATP? Okay, you have 3 lines here. You're going to need them. Hydrogen ions in a high concentration diffuse through an ATP synthase to an area of low concentration. You guys remember the CH word for the production of ATP? Chemiosmosis. What is the form of chemiosmosis where ATP synthase is used? Oxidative phosphorylation. All right, number 1, Calvin cycle. Where does Calvin cycle take place? The stroma. Before the Calvin cycle begins, where is energy stored? ATP. If you want to put bonds, that's fine. What happens to CO2 in the Calvin cycle? It is fixed to RUBP by Rubisco. What are the inputs of the Calvin cycle? Where do they come from? CO2 comes from the atmosphere. ATP and NADPH come from the light reactions. Jack, you checking your work? Number 5, why are the reactions of the Calvin cycle also called light-independent reactions? Light is not a reactant. Circle the letter of each statement that is true about the Calvin cycle. Correct the false statements. The main products of the Calvin cycle are 6 carbon dioxide molecules. Nope. The main products are not 6 carbon dioxide molecules. It is G3P molecules. Carbon dioxide molecules enter the Calvin cycle from the atmosphere. That is correct. Energy from ATP and high energy electrons from NADPH are used to convert 3 carbon molecules into similar 3 carbon molecules. That is correct. The Calvin cycle uses 6 molecules of carbon dioxide to produce a single 6 carbon sugar molecule. That is correct. Eventually, that is correct. What are the products of the Calvin cycle? ADP, NADP plus, and G3P, which does eventually lead to glucose. Number 8, what are the 3 uses for glucose in a plant? Okay, there are 3 here. Cellulose. Let's see. Hey, first unit question, monosaccharide, disaccharide, or polysaccharide? Poly. So you need a lot of glucose to make that. It's like you need a lot of little beads to make a big old Gasparilla necklace. Cellulose is like the necklace. What is cellulose used for? Cell walls. Very good. Another one is going to be amylose. Polysaccharide, monosaccharide, or disaccharide? Poly. This is going to be stored energy. Where do plants typically store it? In the roots. Then we have sucrose, mono, di, or poly. Disaccharide. This is table sugar. This is going to be immediate energy. Down in South Florida, there's tons of sugar cane. You snap open a cane, you got the sugar. It's stored right there in the stem and in the stalk. Okay, guys, before we sign off, I have 4 more minutes. I was hoping for this. I want to go over one more thing with you all before you are relieved of your duties here in class. This is metabolism. This is some of the basics here, so please pay attention. What type of reaction is that? Is energy being absorbed or released? Released. What's higher, the ending or the beginning? So did you end with more or less energy than what you started with? You ended with less, so was energy stored or gained or lost? It was lost. This is exergonic. What type of reaction is that? Stop saying catabolic. This endergonic. What happened? Energy absorbed or energy released? You ended with more energy than what you started with. Energy was absorbed. How about this? Do you recall that? How many reactions are in this? Coupled reaction. There are 4. This is called a coupled reaction where the end of one is actually the beginning of the next. When you guys finish 9th grade, yeah, you're done. That's the end of the school year. That's your product. But it's what you need for 10th grade. So 9th grade is coupled to 10th grade. 10th grade is coupled to 11th grade and so on. What you need at the end is typically what you need for the beginning of the next step. Enzymes. What do you call this area on the top? Active sites. What fits in the active site? A substrate. If a substrate fits in the active site, what comes out? Product. What is this site? No. It's called an allosteric site. If I have an allosteric inhibitor, what does it do to the active site? It changes the active site. Can a substrate like that fit in there? No. Let's look at the opposite. What if I have an allosteric activator? Let's say this needs to fit in there. It can't. You put in an activator, what will it do? It changes it so it can fit. An activator turns on an enzyme. An inhibitor shuts down an enzyme. Last one. Hold on, give me 10 seconds. I'll get you out of here. What if I have a substrate and an inhibitor and they want to go for the same spot? Competitive. Good. All right.